Thank you very much, Dr. Unger. Gus and I are good friends in the United States, and the only thing I understood from my introduction was the American Journal of Psychiatry. So we were, for many years, associated with dueling journals in the U.S., except I just have the opportunity to resign now from the American Journal of Psychiatry, so I have a little bit more free time. So it's delightful to be here again. And I had the chance to give you a lecture yesterday, a talk on my work in BSNP, and now I was invited to give a talk on a completely other topic, which is the hippocampus, which is the research part of what keeps me interested. So this is what the human hippocampus looks like. One of the things that I do in the course of my in the course of my week is contact the medical examiner's office in the city of Dallas, and we collect human postmortem tissue. Initially, I just set out to collect tissue from people with schizophrenia and people with normal controls. The medical examiner, however, couldn't figure out who of the cases that he was looking at had schizophrenia. So I finally had to give him a simpler formula, which was that I would take any case on any psychiatric medicine and any normal control. So that became clear to him, and it provided us with a nice group of brains. This is usually I try to get bloody pictures because the medical students in the U.S. are very attracted by these kind of bloody pictures, so that they'll keep their eyes on the picture. So this is a bloody picture of the hippocampus. This is the anterior part of the hippocampus, and you can see the fimbria under the sheath. This is about the mid-level of the hippocampus in the tail. The hippocampus sits in the medial part of your temporal lobe, and you have two in your brain, one on each side, the right and the left, and I'll show you a few pictures of that as we go on. The hypothesis that I started with when I started looking many years ago at the hippocampus was that psychosis is really a learning and memory disorder, and as a learning and memory disorder, it involves the hippocampus. So I'll tell you a little story first about the hippocampus and about how we got to know so much about it. So that for my whole life, I knew about HM. Finally, HM passed away, and his name became public. So HM, as many of us know, his name is really Henry Molaison. So who was he, and what did studies of him teach us? It was probably about 1952 when Henry Molaison, or HM, had a case of intractable epilepsy. And neurosurgeons knew that if they understood where the epileptic focus was, they could take it out and the person might be cured of their epilepsy. HM, in fact, had a locus, a hippocampal locus, but it happened to be on the right and the left side, both hippocampi. So without thinking about it at all, really, unfortunately, the neurosurgeon went in and took out in one operation the right and the left hippocampus. Whoops. The right and the left hippocampus. So what, in fact, HM, his epilepsy was entirely cured. He never had another seizure in his life. But also he lost all of his capacity to make new memories. It wasn't that he forgot the memories of his childhood. He could remember those memories, but he just couldn't make any new memories. He continued to be involved with his psychologist at the university, and they studied him really for the rest of his life. And then they studied his brain after they were after he had passed away. In fact, the hippocampus was the first piece of incontrovertible evidence that the hippocampus was really the organ in the brain that processes new memories. It's not where all memories are stored, but it's where all new memories are made. And, whoops, this one really wants to go away. So that there were some things that we learned from HM that have been extraordinarily important to us. First of all, that memory is a distinct psychological function. So the neurosurgeons went in and took out both of the hippocampi, and then he lost a discrete function, which was memory. Amnesia, which is what he had, is an impairment of episodic memory, or of hippocampally mediated memory. Amnesia spares short-term memory, so he could remember something for like 10 minutes, like you could tell him a telephone number, and he would remember it and write it down, but he just couldn't remember things for long periods of time. And it really indicated that hippocampus is a core brain structure supporting declarative memory. Because of this, and these clear pieces of knowledge, a lot of both preclinical and clinical scientists went into the study of the hippocampus and were attracted to that, as am I. So the hippocampus is not the only part of the brain that is associated with memory. It's just the most critical part. So if you go into neocortical structures, particularly the sensory cortices and the extensions of the sensory cortices, you see where memory is brought into the brain, the where part of memory and the what part of memory. These are processed very close to the sensory areas. Then they're transported to the parahippocampal region, the regions that are right outside of the hippocampus, and they're still they're somewhat integrated when they go from the neocortical areas into the parahippocampal regions. Then the hippocampus is a region that finally puts all of the stimuli together, all of the new stimuli. For instance, yesterday morning was the first time I walked into this room and everything was new. I understood what the color of the walls were, what the patterns on the ceiling were, what the patterns on the rug were, and my hippocampus was flooded, if you will. If you were doing a functional MRI on my brain, my hippocampus would have been all lighted up. Today when I walked in, because I knew and understood the room a lot from yesterday, I don't think my hippocampus would have even blipped. All of the memories which had been previously put together yesterday were already there just to simply be called up. So the hippocampus has an anatomy, which is a very, very important anatomy, which I'll be talking about a lot today. All of the data from the sensory cortices and the complex sensory cortices come into the hippocampus, into the entorhinal cortex. It's like a funnel. It collects all the sensory motor data from the whole brain. And then there's the main pathway through the hippocampus. It's not the only pathway, but the main pathway is a one-way pathway, starting in the entorhinal cortex to the dentate gyrus, to CA3, to CA1, and the subiculum. And each one of these stations has a separate and distinct function in memory. Oops. Each one of these stations has a separate and a distinct function in memory, and I'll be talking about these a lot, particularly the dentate gyrus and particularly CA3, as these seem to be the most important ones in psychosis. So I've told you a little bit about the normal hippocampus. So what about what would make one think about the hippocampus being abnormal in schizophrenia? There are many alterations that people have reported in the hippocampus in schizophrenia. One alteration is a consistent but a very small 3 to 5% reduction in hippocampal volume. Now, psychosis or schizophrenia is not the only illness where alterations in the hippocampus have been reported. PTSD, some in depression, some in bipolar disorder. So the hippocampus, the processes of memory seem to be altered in a lot of our psychiatric illnesses. However, in people with psychosis, and most of the psychosis work has been done in schizophrenia, most of the people in psychosis, we've demonstrated in schizophrenia a reduction in memory performance, particularly in these relational memory parts and pattern separation. So let me show you a minute what pattern separation is and what pattern completion is. So a memory comes into the brain, and the first thing the hippocampus has to decide is, do I already know this? Do I already have a memory for this, or don't I have a memory? And that kind of decision, if you will, that pattern separation decision, happens in the dentate gyrus. So if you know something, you don't want to be making a new memory, or you'll have new memories in your mind that are repetitive. So if your dentate gyrus says, if they by way of pattern separation, oh, I already have this memory, then you just pull up the memory and don't make a new one. Then you get to the associational cortex, CA3, and you can do pattern completion. If you have a partial memory, if you have a partial memory and need to call up the full memory, you do that too. And these two, it's been demonstrated really that we've been able to demonstrate that both pattern separation and pattern completion are abnormal in schizophrenia. The most interesting thing that I'll show you a little bit of data about today, a little bit more data, is that there's an increase in hippocampal activity. So I'm not even touching this, and it seems to go. You'll see all my you'll see all my data before I get to it. I'm on 4, so that there's an activation deficit there's an increase in hippocampal activity at baseline. So in people with psychosis, young people with psychosis, you'll see an increase in their hippocampal activity. And then if you give them a task to do, you'll see a deficit in the activation of the task. And it's recently been shown, Stephen Heckers has just shown, that those correlate with each other, suggesting that the increase in hippocampal hyperactivity is really what's causing this activation deficit. There's a decrease in neurogenesis in dentate gyros. We don't quite know what that means yet. But this is just a list of all the things that are abnormal in schizophrenia. Now, how would anyone put that together? So it used to be that before I had thought about it very carefully, all of us, when we study the hippocampus, would take out the hippocampus, freeze it and then pound it all up into little powder and mix it up, so that when we examined the hippocampus, we would examine a homogeneous part of the hippocampus. Well, you can imagine from everything I just told you that that's really the wrong way to study the hippocampus, because the hippocampal function is dependent on its anatomy and dependent on its subfields. And it wasn't until we started looking at hippocampal performance by subfield that we got to understand. Let me see what's here. I'm going to show you a little bit of information about the hippocampal hyperactivity, which is a characteristic of schizophrenia, probably a characteristic of all of psychosis. These are some data, really, that I showed you yesterday. I showed you a lot of anatomy from the BSNP study. This is another analysis of the structural of the resting state data, resting state fMRI data, and the blue areas that you see are the areas that are abnormally reduced in schizophrenia, where resting state activity or baseline basal activity in the brain is reduced in schizophrenia. What you see here are there's two areas, one on the left and one on the right, and where resting state fMRI was increased, and this is really the left and the right parahippocampal hippocampal cortex area. So that was one of the ways that we knew the hippocampal anatomy. Hippocampus was hyperactive in schizophrenia, and here are two others examples. Here's the hippocampus as it, this is flipping all around. I must be doing something wrong here. Here we go. This is the hippocampus as it, this is the hippocampus as it sits. I'm not going to point anymore. I'm just going to refer you to the yellow arrows. The yellow arrows is the anterior part of the hippocampus, and the black arrows are the posterior part of the hippocampus. It lays in the medial temporal lobe, just off the surface of the medial temporal lobe, abutting the lateral ventricle. The hippocampal hyperactivity in schizophrenia is really located in the anterior part, right around the yellow arrows. And we've demonstrated on the left side of the screen using PET, using PET 015 water. And then we've demonstrated also on the right where using a technique called the vaso technique, where we could distinguish subfields. So the dentate gyrus has about an equal in schizophrenia, which is in orange, and in the normals, which is in pink. The resting state hippocampal blood flow is normal in dentate gyrus, but increased in CA3 and CA1. So I wanted to use the hippocampus. This is a picture that you just saw of the hippocampus. We sliced through the hippocampus and did some microdissections. So we actually got tissue from dentate gyrus, got tissue from CA3, and got tissue from CA1. And I set out to look at, whoops, you saw the punchline. I set out to look at hippocampus by subfield, looking at markers of inhibitory function and looking at markers of excitatory function. As it happened, what you see on the left is the human hippocampus, and it's stained for NMDA receptors, so that NMDA are the excitatory receptors, which you see in black, and the curved U-shaped object is the dentate gyrus. Right coming out of the dentate gyrus, I have to show you this, whoops. Coming out of the dentate gyrus is CA3, and this area here is CA1. So I really took the dentate gyrus, then dissected away CA3, dissected away CA1, and got tissue that was really enriched for those areas. So the first thing we saw, now mind you, I'm looking for a reason for hyperactivity. So that hyperactivity could be caused by a decrease in inhibition, so we could find decreases in glutamatergic markers, or increases in excitatory markers. So the first excitatory marker that we looked at was the gluin-1. Gluin-1 is a subunit of the NMDA receptor, and it's the necessary subunit. So if you find NMDA receptors that don't have any gluin-1 subunits, you know that they're excitatory receptors that don't work anyway, so that they're good markers of the excitatory glutamate receptors. And we also looked at GABA. We found GABA unchanged throughout all these dentate gyrus areas. GAD65 and GAD67 were unchanged. But what we found with gluin-1, whereas we had been expecting an increase, we saw a decrease. You'll see right dentate gyrus CA3 and CA1, those values are spread out along the horizontal axis, and if you look at dentate gyrus, you can see in orange the samples from the schizophrenia brains, and in green the normals from the values from the normal brains, and you can see that there's a significant decrease in dentate gyrus. So the only way to interpret this was really that for some reason there was a molecular lesion in the dentate gyrus. So now I've redrawn the anatomy of the hippocampus in humans, and this would be a psychotic hippocampus where perhaps, this was a hypothesis at the time, there was a molecular lesion in dentate gyrus so that there was a reduction in dentate gyrus. Somehow all of these slides are like going by themselves.
Yeah. There's some problem.
Okay. I'll just work with it. So our hypothesis is that there was a lesion in the dentate gyrus, that the mossy fiber pathway, which you see here in the dotted line, there was a reduced activation. And then for some reason, which we know more about now than we did before, the response of CA3 was a hyperactivity response rather than a hypoactivity response. There was a supersensitivity by way of metaplasticity in CA3, and that increased the activity in CA3. That increase was projected to CA1 and projected to subiculum. And then what we were suggesting is that there would be simply too much a hyperassociation in CA3, so that CA3 would actually make mistakes of association, so that delusions, maybe hallucinations, would be activated by mistake. Those of us who have spent lifetimes with people with psychosis and schizophrenia understand that they don't really have new hallucinations every day. They don't have new delusions every day. That if you really look at the symptoms of hallucinations and delusions, they look a lot like memories. They look a lot like abnormal memories. So then we set out to look at inhibitory and excitatory markers, both in CA3 and in CA1, and I know you've seen this already. But in CA3, we found changes both in this gluin-2B, the gluin-2B receptor, and we found changes in PSD95. So just in CA3, in dentate gyrus these were normal, and in CA1 they were normal. But in CA3 we found that in the people with schizophrenia, they had elevated gluin-2B over gluin-1 NMDA receptors. Now, the significance of them is that the gluin-2B type of NMDA receptor is the supersensitivity kind of receptor. So for every activation of the NMDA receptor, if it contains a gluin-2B subunit, you get much more of a response. So this would suggest that these NMDA receptors are the supersensitive type. And PSD95 is one of the proteins on the postsynaptic side of the receptor that is increased with LTP. So if the NMDA receptor is hyperactive, more PSD95 comes to the postsynaptic membrane in order to mediate that supersensitivity. So both of these both of these markers would suggest that there was increased activity in CA3, which is really paradoxical. It was what we had hypothesized, but it was paradoxical to the effect that we found in dentate gyrus. Since that time, I've had a chance to scour through the preclinical literature, and actually people in the preclinical literature have noticed in the past that there's a special characteristics of the mossy fiber CA3 synapse, and that this particular synapse is very, very sensitive to changes in the efferent flow of glutamate. So if I had known that before starting out, this wouldn't have been any surprise at all, because we were thinking, because we had biochemical evidence then in the human tissue. Because we had biochemical evidence in the human tissue of hyperactivity in CA3. I paired up with a friend of mine, Lynn Selleman from Yale, who does a beautiful Golgi in human tissue. So this is Golgi staining of the CA3 in the hippocampus, of the human hippocampus. So this isn't a rat, this is a human, which makes it even prettier. Oops. This is a pyramidal neuron in the hippocampus, a human pyramidal neuron. This is the cell body area. This is the apical dendrite right here. And these are the axonal elements at the bottom. So this is from some normal tissue. This is a same example from tissue from a person with schizophrenia. There's two boxes you can kind of see. These are the boxes. These are not natural, but we put these boxes on them on the apical dendrite. So this apical dendrite, after its first bifurcation, this is really the area here where the mossy fiber pathway comes from the dentate gyrus and synapses on the pyramidal neuron. Here's the pyramidal neuron. This one is a little blotch here. These black blotches are just blotches of stain, and they're not meaningful. This is the pyramidal neuron in the person with schizophrenia. This is the first bifurcation, and this in the person with schizophrenia. This is the area where the mossy fibers come in. So you can and here is just an example of the areas where the recurrent collaterals come. Now you can see that these areas here between the person with the normal control and the person with schizophrenia, these you can just see it with your eye that they're exactly the same. At the impact of the mossy fiber pathway, however, you can see the normal apical dendrite here with a few little boutons, with a few little places where the mossy fiber pathways would synapse. But then if you look at the same area on the apical dendrite of the person with schizophrenia, you can see just a wild overgrowth, if you want to put it that way, wild overgrowth of synapses. So the basis of the hyperactivity, actually I should have just left it, the basis of the hyperactivity in hippocampus really is founded on this perfusion, the increase in the number of synapses. And the increase in the number of synapses in the mossy fiber CA3 terminal area in people with schizophrenia versus normals is really reflected in the change in the NMDA receptor I showed you and the change in the PSD95, and we also saw some BDNF changes. And those are really confined to CA3. They're not located in CA1, although the hyperactivity is perpetuated downstream, so it's CA3 and CA1. In fact, when you do human imaging studies, you see the clearest signal of hyperactivity in CA1. And the dentate gyrus at the same time, it has reduced gluin-1 and probably has reduced activity as well. So this shows that if you look carefully at the human hippocampus, you can find differences by subfield that then would explain the hyperactivity. So we know the summary of what I've just told you, even though you might want to miss all of the data pictures I showed you, is that there's increased hippocampal blood flow, and that's consistent with increased neural activity. In postmortem human tissue, there's an increase in synaptic strength at CA3, and we really suggest that that's influenced by the reduced glutamate signal in dentate gyrus. And then in postmortem brains, we can see increased spines, and we can see terminal insertions, increase in terminal insertions in CA3. I hadn't before. I mean, I've looked at a lot of human postmortem brains, and I've seen a lot of subtle changes in human postmortem brain, but never have I seen a change in synapses that was so prominent and so unmistakable as what we found in CA3. The understanding, although it's just a hypothetical understanding, and I haven't demonstrated that in any way, is that that psychosis is associated with hippocampal hyperactivity, and that hyperactivity in the schizophrenic hippocampus generates false memories, and it generates memories with psychotic content. We haven't yet gone into a person with a hyperactive hippocampus and psychotic symptoms, been able to decrease activity in the hippocampus and then see a change in psychosis, which is what we have to do in order to demonstrate this. But we're on our way to doing it. And then there's a lot of things you can't do if you're looking at human tissue. We only get human tissue as it comes to us from the medical examiner's office. We can't make a perturbation and then look at hippocampus. So it was of some interest that we saw a mouse with exactly the perturbation that we were interested in, because it was an exact duplicate of what we had found in schizophrenia. And this is one of Tamminga's very interesting mice, where he deleted gluin-1 in the dentate gyrus. So the data that I have shown you so far, and the hypothesis that we've made of it, is that there's a lesion in the dentate gyrus, and that lesion causes the hippocampal hyperactivity. It could be that there are other neurochemical lesions within the schizophrenic hippocampus that makes them vulnerable to this. That would be what we would think for sure. But that increases synaptic strength in CA3. Oops, I'm done with this. Anyway, we found a mouse. Oops, I'm going to have to tell you one more story about transcription analysis before I start with the mouse. We took these regions, dentate gyrus, CA3 and CA1, and we did a transcriptome analysis, and we found an interesting overlap between what anybody would call memory genes and what we found in the CA3 psychosis genes. So we found 336 abnormally transcribed genes in the schizophrenia tissue, and this is 297 genes that David Sweat has defined from memory studies in mice. So there's a mice that he takes mice and he teaches them a task, and then he samples their hippocampus before and after the memory event, and through a technique like that, he's really defined for himself in the mouse 297 genes that he calls memory genes. So there's really a substantial overlap in these two gene sets, one which is a CA3 psychosis gene and another one which is a CA1 memory genes, and it's at highly significant level. So there is, from a genetic point of view, we've identified already, although we haven't gone further with this yet, we've identified already an overlap in the expressed genes in hippocampus between the human psychosis genes and mouse memory genes. So let me show you more about the mouse, because there is a specific mouse. This is Susumu Tamminga's mouse with a specific deletion in gluin-1 in the dentate gyrus. And what I'll show you in this mouse is that this is almost an exact it's almost exactly the same in many different ways as we see human psychosis. And this shows really that the change in the deletion of gluin-1 in the dentate gyrus can cause a whole system of changes downstream. And I'd like to show you those in a minute. So we study the control mouse and the knockout mouse inside of that red box. And the control mouse is a regular wild type mouse, and the knockout mouse is a knockout with the gluin-1 dentate gyrus specific knockout. So what you can see here, I teamed up with an electrophysiologist to take this mouse and put a probe into the dentate gyrus and put a probe into CA3 to see in a situation where gluin-1 is knocked out only in the dentate gyrus, what do you see? The first thing you see is a decrease in the probability of glutamate release at the mossy fiber CA3 synapse. So at the mossy fiber CA3 synapse, in the gluin-1 knockout, just like you would expect, you see a decrease in glutamate release at the mossy fiber CA3 pathway. Okay. Oh, I got something better now. Thanks a lot. Yeah, there's a decrease in gluin-1 release. So we know from this mouse that if you delete gluin-1 in the dentate gyrus, you'll get a decreased release of glutamate onto the CA3 neurons. And then the question is, what happens at the CA3 neurons? The first experiment that we did was to look at what was the NMDA-AMPA ratio. It's a kind of a usual experiment because usually NMDA and AMPA don't change together. One of them changes or the other one changes. So we found the NMDA-AMPA ratio the same between the knockout and the wild types. Every time, this is all in the mouse. So we saw no change. Needless to say, we decided to go on anyway, and we decided to look at the AMPA receptor and the NMDA receptor separately. And that was fortunate because you can see in the gray circles here versus the golden circles. The golden circles are the knockout. The gray circles are the controls. That the knockout mice had in CA3 at the mossy fiber, the synapse, had a huge increase in the number of AMPA receptors. Also had a huge increase in the number of NMDA receptors in the knockout mouse compared to the wild type. So this you could see in the electrophysiology, the electrophysiologic responses that were the parallel of this increase in AMPA and increase in NMDA. So that with just the deletion of gluin-1 in the dentate gyrus, we had these huge increases in AMPA and NMDA receptors within the CA3. So what should happen is that we should be able to find markers in the mouse of hippocampal hyperactivity. And in fact, we did. We looked at CFOS. We looked at the number of CFOS-activated neurons in CA3. Now we already know that there's and this is still in the mouse, we already know that there's an increase in AMPA receptors, an increase in NMDA receptors, and then we found an increase in we found an increase in the number of CFOS activated cells. Here is the data from CA3, here is the data from CA1. So you see, you can see that the increase in CA1 is lower, but it's still a highly significant increase. I don't think you can probably see them from the audience, but here's a control tissue, and here's the knockout mouse, and you can see the number of cells that are stained, control tissue, and the number of cells that are stained there. Now, what is interesting is in the mouse, this increase in the number of activated CFOS cells, CFOS-containing cells, which is the marker of hyperactivity, is not all over throughout the whole hippocampus in the mouse. This is the dorsal part of the mouse hippocampus, which is really the posterior part of the human hippocampus. Really, it's only in the ventral part of the mouse hippocampus, and that would be the correlate of the anterior hippocampus in humans, where there's an increase in these CFOS-activated cells, and also in that same, only in that same area are the cells increased in CA1. So what I've shown you so far is that from a little change in a mouse, which is the deletion of CA1 in dentate gyrus, we get really substantial increases in CA3. In fact, it was almost just by accident that the person who was doing the CFOS changes said, it was one of my technicians, and she said, Carol, when I've looked through the mouse brains, I find pockets of increased CA3 activation throughout the neocortex or throughout the rest of the mouse brain. Are you interested in those too? So I said, sure, of course. So what the data that I just showed you were data from CA3, CA1, and the subiculum. So she had noticed, actually, both in the nucleus accumbens, which is a part of the striatum, in the medial prefrontal cortex, not the dorsolateral prefrontal cortex, but in the medial prefrontal cortex, just anterior to the cingulate and involving the cingulate, we saw increases. And then in the amygdala, which is intimately related with the hippocampus. Not only were there increases in these three areas, but the connectivity between CA3, CA1 was remarkably increased between all of these areas. And if you look at these areas carefully, they really represent the limbic system. So in this case, in this mouse model, we made one lesion in the mouse model, and that was just to delete gluin-1 in the dentate gyrus. And when that's deleted, we got activation of this whole downstream circuit. We hadn't done anything in these downstream circuits at all. So that the abnormality that we noticed in each one of these areas really is due to the abnormality in the dentate gyrus. So now we wanted to see whether this made any difference for the mouse. And we've done a number of different behavioral analyses in the mouse, and we really noticed that the gluin-1 dentate gyrus specific knockout mouse has animal behaviors that are traditionally associated with psychosis. And this just shows you one of them, which is a social behavior. And first of all, I'll just draw your attention to the cellular activity, and this is all done in CA3. First of the wild type control. So this is CA3 activity in the wild type control. This is the knockout mouse. That's the brother or sister of the control. And you can see a significant increase in CA3 cellular activity. And if we put in an inhibitory dread into CA3, and if we decrease the cellular activity in CA3, you can see that we can drive that cellular activity down to normal. And what happens then in social behavior is the wild type control has a normal amount of, this is novelty recognition behavior of another mouse. You can see a normal level here. The knockout control, this is the dentate gyrus control, which is really the what we would call a reverse translation model of schizophrenia. You see a decrease in social behavior, and you can restore that if you just inhibit the cellular activity in CA3. So concluding from all of these data, what I talked about yesterday was really the segmentation of psychotic disorders into individual diagnoses. And what I've been talking about today really has been a potential hypothetical mechanism for psychosis. And this would be, there's no reason to think that the generation of psychosis in any psychotic disorder would be different. And one could really see through mechanisms like this. One could see one could see mechanisms of psychosis all throughout brain. One of the things that we're really anyway, I'll talk about that in a minute. So that we would, at least our current working model is that this hippocampal abnormality that I've talked about may be a common pathophysiology to all of psychosis. And then I've said before that we really think that the hippocampal hyperactivity may be involved with making loose associations, with making aberrant associations, some with psychotic content. Clearly, there are many areas involved in, many different brain areas involved in psychosis. I haven't even said anything about the dorsolateral prefrontal cortex, and it's not an area within the human schizophrenic brain that I really have studied much, because there's plenty of people studying that already. And we don't know anything about how these various areas that have been already identified as abnormal in schizophrenia. We don't know how any of these areas are tied together. The dorsolateral prefrontal cortex, the thalamus, the cerebellum, the hippocampus. That's probably tomorrow's story, but we're interested in how it unfolds. These are just a list of my people who've actually worked on the study. And then there's the question that comes from this, which is like, is this ever going to come to anything practical? Mice being good for what they're good for, clinicians aren't really awfully interested in it. What would it make us think about? The last point here, we could really, we could have the idea that hippocampal inhibition was a good idea in people with schizophrenia. So theoretically, a drug like levetiracetam, or in the United States, levetiracetam is called Keppra. It's one of the most widely used of the anti-epileptic drugs. That we should be able to take levetiracetam, administer it to people with schizophrenia and with poor memory, and we should be able to show that the memory improves and that perhaps even the psychosis is reduced. I'm not sure that if somebody has a lifetime of psychosis, so their hippocampus has been working, and the psychotic memories have been exported from hippocampus, I'm not exactly sure that you would see a decrease in psychosis. You might see a decrease in the new production of psychotic ideas. So if this levetiracetam was a drug that was given very, very early in illness, and in fact, early treatment is one of the goals of modern psychiatry, at least in the United States, levetiracetam might be a drug that would really work. We're going to really try the administration of levetiracetam first in our mice and see if we can cure their CA3 hyperactivity, and then we'll be giving levetiracetam to patients to see what happens. So with that, I think I will end and see if there's any questions. I have to say that the second clicker worked really well. Thanks a lot.